Okay, folks, if you don't mind, I'm going to try to get going, and we'll try to get through as much of the lecture as we can today. Then you're going to help me out at the very end of today's lecture on trying to set things for your midterm. So we'll leave about 10 minutes at the very end of today's lecture on that. So we're going to continue on a little bit with what we've been looking at in the gut microbiome, and we're going to go back and revisit the important role of fiber. And then we're going to change gears just a little bit. We're still going to be looking at the gut, but in a slightly different way. And again, I want to make this relevant for all of you in the sense that in terms of looking at diet, we've been looking at things that directly influence the gut microbiome, but now I want to look at things in a slightly different way and take you through a paper that came out at the end of October last year related to your gut health as well as potential carcinogens. And this will also set up things for next week's tutorial as well. So we're going to take a look at the importance of fiber, and actually very, very surprisingly, the fact that if you are looking at potential effects that go beyond just your physiology into generational effects, so your children, biome as well. And then we're going to talk eventually in the second part of today's lecture a little bit on carcinogens. Whether you're aware of it or not, the types of carcinogens that we're going to be mentioning in class today are actually considered either as class 1 or class 2A carcinogens, which means that in many ways they're just as bad as nicotine, they're just as bad as asbestos, they're just as bad as any other normal carcinogen that you can think of as well. For those of you who have the little packages in front of you, could you open those up now? Feel free, by the way, to share if you are near anyone. So if you take a look, what is the how much fiber does that actually contain? Sorry, 20%. But I want you after today, because this is actually the really important thing. When you're reading through these labels, which all of you are doing because you're in university, you care about your health, and when you read things related to things like 20% fiber, 20% fiber means nothing. Actually, it doesn't mean a single thing in the context that I'm going to be showing you a little bit later on today. So we're going back to looking at the gut microbiome. One of the huge researchers that's in the field, and you don't have to memorize this quote or anything, but diet actually has a tremendous impact on you, whether you believe so or not. Diet is one of the most powerful tools that we have, and this is Dr. Sonnenberg down at Stanford University. And one of the things that we know is true, and one of the reasons why people are so interested in fiber right now, is that fiber has been shown to influence the bacteria, because things that you can't digest, your body produces a number of enzymes that help in the digestion of different types of fibers, but certain types of fibers that our bodies can't digest, we don't have the right enzymes, things that are found in vegetables, whole grains, and in other types of foods. The bacteria can digest those, and the bacteria can directly influence what happens, especially in the large intestine. And this is something that we're going to come back and take a look at in a slightly different way. So this is a slide I'd like you to get the gist of. Why do we talk about fiber? How does it impact us? How does it relate back to the lectures that we've had earlier on the mucosal firewall, the gut firewall that we looked at the end of the last lecture? This is a very, very small study. This is the same type, by the way, of snack bar that you're eating now. And this snack bar is sort of the thing that everyone likes eating right now for a variety of reasons. A very small study that was published in 2015 where there were 21 healthy individuals around your age, a little bit older and a little bit younger. If they are eating one of these daily snack bars, which contains roughly 21 grams of fiber, and again, the numbers not being important, they were found to significantly increase the number of bacteroidetes types of bacteria, and they also decrease the number of firmicutes. So if you ever wonder why there's such a big change now in sort of this fiber-rich diet that involves snack bars similar to what you're eating, part of it is because a number of these studies are coming out. Mind you, the food companies are using this in a different way. The types of fiber that they're using there, and these are not commercial snack bars, the fiber there is different than the ones that you're consuming right now in your snack bars. And again, just as a reminder for you, because this is an important idea, that your gut microbiome is always in a balance. There's always a balance from the time that you were born to the time that you reached about 2 to 3 years of age, that you had a balance between the bacteroidetes as well as the firmicutes. And throwing that out of balance lead to things like CDAD or Clostridium difficile associated disorders, or it leads to improved health. And we know that increasing the number of these bacteroidetes and reducing the firmicutes has also been shown to be beneficial to individuals as well. So our fiber can actually directly affect us. I'm going to come back to this point about mucus a little bit later on. This is an important point as well. So your recommended fiber intake is somewhere on the order of about 25 to 38 grams of dietary fiber every day. You are far below that, okay? And we're talking about real fiber, not the processed types of fiber that you're looking at right now on your food labels. What that fiber is on your food labels is actually complex carbohydrates. It is not the type of cellulose that you find in vegetables. It's not the type of fiber that you would normally associate with raw fruit. It is just a complex carbohydrate. Can your body break down those complex carbohydrates? They can. And that's not the type of fiber that we're looking at. And our genome has a number of really good digestive enzymes that can break down carbohydrates into simple sugars that our body likes to uptake. And the types that are in those snack bars are not the ones that cannot be broken down. Your body can actually break those types of fibers down. They're still labeled as fiber, but probably they're not as good for you as getting things in your diet through fresh fruit as well as vegetables. So true dietary fiber, not the types that are on these labels, are ones that your body cannot produce the enzymes for. Your body doesn't have the enzymes. Our genome is limited. We don't encode for the types of enzymes that can normally break things down like cellulose. And as a result, when we are taking in those complex carbohydrates, we can break those down, but these other polymeric molecules that our bodies cannot break down are really only found in things like fresh fruit, vegetables, etc. And one of the reasons, again, we're interested in this is because your gut microbiome, all of the bacteria in your gut, they can produce enzymes that can break down these larger polymeric molecules. In return to doing this, as they start to digest this down, they will convert these into short-chain fatty acids. They can be used as fuel for other intestinal cells, and they will also help in the production of mucus. Now, go back, think a little bit about what this implicates, right? So they are producing these short-chain fatty acids, which can then act as fuel sources for the intestinal cells. These intestinal cells that they're in contact with in your large intestine are now going to be able to produce mucus. Is that a good thing or a bad thing? Any ideas? 50-50 guess. Good thing to have more mucus in your gut or bad thing? Yes. It's a good thing? Right, so having a thicker mucosal layer, thicker mucus, not mucosal layer, thicker mucus is important, and it's one of those protective mechanisms that we take for granted. We don't really think about mucus as a protective type of barrier, but in fact it is. And having this extra source of energy that allows your gut to produce more mucus is actually beneficial for a gut in the long term. And so are there some benefits? And we're going to come back to this again next week when we start to talk about colorectal cancer, but potentially there's a lot of benefits to having excess energy being converted by your gut microbiome as they start to process some of these polymeric molecules that our bodies just don't have the genome to produce the enzymes to digest. So again, I'm going to go back and talk a little bit about experimental studies that were done on humanized mice. I'm not going to go through this in a lot of detail because I went through some of this in the previous lecture. So in order to see what are the long-term consequences when you start affecting the gut microbiome because we have fewer fibers in our diet, what is the long-term consequence in humanized mice? And again, we're using humanized mice in the sense that the gut microbiome is from a human. It is still the mouse system, but all of the gut microbiota are actually taken from a healthy donor male. So again, we know that as you reduce the complexity of what these dietary fibers look like, and we start reducing the overall fiber intake, that one of the things that happens is our gut microbiome, not unexpectedly, starts to change. And as we start having a diet that is low in these microbiota accessible carbohydrates, or MACs, in other words, a Western style diet that is lacking in fiber, then we start to lose specific types of gut microbiota. Again, this high MAC diet is one that is rich in plant polysaccharides, ones that are not easily digestible by our digestive enzymes, but they are by the gut microbiome, and these come from a variety of different sources. And the low MAC diet that we looked at previously has carbohydrates that are relatively easy for our bodies to digest, and there's nothing left for the bacteria to be able to digest by the time it reaches them. So again, this 3-week-old paper was actually really, really surprising. Not surprising in the sense that the gut microbiome is changing. That wasn't so unexpected. The fact that if you take parents and you breed those parents that have been on this low MAC diet, their offspring also have a reduction in the gut microbiome or the gut microbiota in terms of their diversity as well. So there were a couple of things that I neglected to mention on this last slide, and I think I took you through the model in the last lecture on Monday. And I wanted to go through this again with you in a slightly different way and to make sure that you understand this particular data point. So these initial results that they did was to take these humanized mice, so mice that were had an oral gavage of the gut microbiome from a normal healthy male from the San Francisco Bay Area. The gut became colonized by the human gut microbiome, and then they were put on a really rich fiber diet or a high MAC diet for 4 weeks, and then they were switched over. Either they stayed on the high MAC diet or they were switched into a low MAC diet for about 2 months or 7 weeks, and then they were switched back. And one of the things that they wanted to look at was to see diversity. And diversity can actually be looked at in a couple of different ways. One measure is to look at this index known as the Principal Coordinate Index, or PC1. And all this says is that at the very bottom down here, there is like almost no diversity. It's kind of like going to the Oscar show and seeing like all the leading actor nominees. Very low diversity down here. And up here, we have a lot of diversity. Okay, so if you're taking a look at the PC1 as one index of diversity, what they're seeing is if we are giving these mice a really high fiber diet, a high MAC diet, they have a lot of diversity in the bacteria that live in their gut. If we switch them, and we switch this group down here, and this is this switch that they have over here, if we switch them into a low MAC diet, so a westernized diet, and then we go back and we probe their and we probe the gut microbiome. And one of the ways that we can probe the gut microbiome is by taking the bacterial species, isolating what is known as the 16S ribosomal RNA from each of the bacteria. Each bacteria has a unique 16S ribosomal fingerprint, and then run all of those. What we'll see is that there are fewer and fewer bacteria that have unique 16S amplicons or 16S ribosomal RNA, which is what you're seeing here. These all of the 16S ribosomal RNAs, the numbers of unique fingerprints of these 16S ribosomal RNAs in the fecal samples have all disappeared. But they stay in the individual or in the mice that have this high MAC diet. So what happens then if we take a look at these points over here. So they were on this low-mac diet for a while, and now we do this diet switch. We put them back on high fiber. You'd think, wow, it must be a good thing, right? So we know that we can switch our gut microbiome, and we've switched it in a bad way. We've lost a lot of diversity. We've brought it all the way down to having low numbers of unique fingerprints that we can identify through the 16S ribosomal RNA. But when we switch them back to a high-mac diet, do they stay at the control levels? Do we get them all the way back to where we would normally expect them to be? And it turns out that no, we can't. That even if we start putting them back on an enriched diet, that we never retain or we never get back the diversity that we started off with. And so what this suggests is that these are long-lasting changes that occur in the gut microbiome. So the diet switch is, again, not so surprising, but the fact that you can't recover fully the gut microbiome unique fingerprints as determined by 16S RNA was kind of surprising, to be honest. And this is why these levels, when we switch them back to a high-mac diet down here, when we switch them back, they don't go all the way back to the levels that we would normally expect them to be. Do you have any questions on this before we go on? I know some of you had some questions at the end of the last lecture. Yes, question? So yes, this is suggesting that there is limited recovery and that we lose a lot of the diversity and we can't recover all of it. And the way that we detect for diversity is through unique bacterial fingerprints using this 16S ribosomal RNA. One thing that you should make sure that you know from this slide, how do you test for bacterial diversity? You run all of them, you see how many unique 16S ribosomal RNAs there are. If we start out with 1,000 unique 16S ribosomal RNAs, we should still have about 1,000. But after a low-mac diet, it might drop all the way down to maybe about 50 unique or 100 unique ribosomal RNAs. So again, this is a really busy slide, and I'm not going to ask you to memorize all of the diets, all of the data from these different types of diets. But what this is showing is that if we take that same humanized mouse, and then we put them on this low-mac diet, and then we look at the pups that are born, and we put the pups on a low-mac diet, so the mother or father has been on a low-mac diet, so low fiber amounts. They have pups that are born to them. The pups themselves are put on a low-mac diet. And then after they are weaned, after they stop taking up the mother's milk and they are no longer weaning, if we put them on a high-mac diet, can we recover any of that diversity? And again, using the same sort of index that we saw earlier, that principal coordinate access to look for diversity, what this is really showing, and you don't need to follow all of these different data points. This is G1 if you're interested in going back later. The major points are down here at the bottom on these bullet points. G1 is generation 1, so the first set of pups that were born. You do the same experiment for generation 2, so you take these pups on a low-mac diet, and then you cross them and you get the second generation, the third generation, and the fourth generation. And what you're seeing is no matter what happens, when you put them back into a high-mac diet, there are a couple of things. One, that over time, that through the different generations, the level of diversity that they're beginning with is going further and further down. And that when we put them back into a high-mac diet, the recovery phase is actually lower and lower. So it gets worse. It gets amplified over generations. From generation 1, it was bad. But for generation 4, there's almost no diversity left by the time they start. And when they put them back to a high-mac diet, the small increases that they saw in generation 1 in the top bars in yellow are almost gone as they take a look down here in purple. So we don't see a recovery. We don't see this rebound going up in generation 1. In generation 2, there's a small increase. Generation 3, there is no increase whatsoever. So this is a long-lasting effect. The longer these mice are kept on this low-mac diet, the worse off that they are. Do you have any questions about this really quick before we go on? And again, how are they measuring these changes in diversity? What sort of tests are they running? So this is, again, the same 16S ribosomal RNA. They're taking all of the gut microbiome. They're taking a look at the 16S RNA, ribosomal RNA from each of the different bacterial species, and they're seeing less and less of these bacterial species. So switching to this high-MAC diet only allowed for small recovery for a very small number of taxa. And again, don't worry about the numbers. Don't worry about any of the ratios. We don't get any kind of big change. It's almost undetectable by the 4th generation. So the small amounts that we saw in the yellow bars or the yellow circles in the previous diagram are almost gone by the time we reach generation 4 that you're looking at here. When we put them back on high MAC, it's almost non-existent. And again, one of the things that is surprising that they start to lose some of these different types of bacteria faster than others. So they start to lose, for example, the one particular taxa much faster than the other taxa. The Clostridiales, for example, tended to be lost slower than the bacterioid type of taxa that I'm showing you here. So different types of bacteria are being lost. All of them are being lost, but some are being lost at a faster rate than others. So they wanted to know, is that really the end of the road? So if this is the worst case scenario, that every subsequent generation, when you're on a certain diet and you're in that sort of setting, that the generational effect is going to be amplified and we can't recover. Every generation, it becomes harder and harder to recover any kind of diversity. They wanted to know what happens at the end of all of this. So is this like you can't do anything for these mice at all. So remember, this is the 4th generation. The 4th generation mice that I just showed you, when we switched them to a high MAC diet, nothing can be done. The diversity does not come back. However, they wanted to know, these 4th generation mice, if we put them on a high MAC diet for a while, again, no recovery to begin with, if you take them back to the original state, so this is 4th generation, so this is the great-great-grandchildren of the first experiments that were done. If you take them back to the original state where you did a oral gavage on the control mice, and you put back in the original gut microbiome through fecal transplant, similar to what we talked about before, does this help them? Is there something messed up with their gut, or is it really something that is unique to the gut microbiome? So when they receive this fecal transplant, surprisingly, they go all the way back up to the diversity that they started out with. So it's really not all lost. Your tissues themselves are not really affected. Something is happening to your gut, which we don't fully understand, but the diversity can be recovered as long as you have the right sort of transplant system in place. And this was sort of the surprising thing about this, that this fecal transplant actually allowed for almost full recovery, back to what you would expect in terms of diversity. The last thing I'm going to mention on this particular slide is this. They are also using a mathematical formula, which I put on another slide earlier on. You don't need to worry about this, but a Shannon index is often used in ecology and in evolution to also look at diversity. So one way to look at diversity is to look at the principal coordinate axis, and another way is to use this particular index, which is known as the Shannon index. By both measures, what we're seeing is that after fecal transplant, the diversity comes back, okay? Do you have any questions about this before I go on? And again, you don't need to worry about how the Shannon index actually is calculated. There is a mathematical calculation for this, but it's not all lost. You can recover a lot of function, and that's an important consideration. Do you have a question? Yes, good point. So in order for them to recover and to actually get back to where they need to go, you can't keep them on a low MAC diet. You need that high MAC diet and this transplantation for this to occur. Great point. Thank you. Do you have a question as well? Okay, also same question. So you need that high MAC diet. You can't have this recovery with a low MAC diet and fecal transplantation. You need to give them something to make sure that they're staying in that healthy state. Do you have any other questions before we go on? No? In Monday's class, in Monday's tutorial and in Monday's class, we're going to be switching gears just a little bit. All the things that we talked about, by the way, are things that are up to you. They're all voluntary. You decide whether or not you want a high fiber diet. You decide whether or not, in most cases, where it's financially and economically feasible, you decide to do all of these different types of things. How many of you remember late last year, October 26th, 27th-ish of 2015, where you were on that particular day? October 26th, 25th. Where was I on that day? Maybe you remember, maybe you don't. I happen to remember that day. I remember that day for one reason, and it's because of this particular slide. Some of you have come to visit me in my office over in Human Bio, and you'll know that we sit right behind Sid Smith. And one of my favorite things to do is to walk through Sid Smith and go out to St. George and visit one of those fine eating establishments that they have along St. George, whether it's one of the food trucks or the hot dog stands. And I love hot dogs. And I was there buying a hot dog, and I was halfway through this hot dog when a student came running up to me at the hot dog cart and said, "Dr. Ju, have you read this new paper that came out in Lancet saying that meat is bad for you? You should stop eating that right away." And of course, I completely ignored the student. I said, "Thank you. Thank you for the great advice. And, I'll stop eating this hot dog when I'm done." And I started to go on. But actually, because they were so insistent, I went back to the office. I pulled open the Lancet Oncology article that was making headlines everywhere. It was trending on social media and everywhere else. Basically, what this article that you'll see different pieces of starting on Monday is suggesting that meat, red meat, and processed meat, which are different, and I'm going to show you some of that today. Red meat and processed meat are actually considered to be carcinogens. If you eat a certain amount of meat every single day, that you are putting yourself at risk or the same risk of developing cancer as smoking or drinking alcohol or having a viral infection. And that may be surprising to a lot of you. But I also want you to be thinking about it this way. So a lot of you read these labels and you read things like, oh, fiber, good for you, not really sure why, not really sure what fiber really means, but hopefully now you have an idea. But I also want you to be thinking about it this way. Why does meat, I'm not talking about processed meat, I'm not talking about things that have chemicals added to it. Okay, we'll get to that eventually. And that's actually the worst carcinogen. So having processed meat, which was all over the newspapers as bacon, so stop eating bacon, it's bad for you. Why is meat, red meat, bad for you? Have you given any thought to that? Why is red meat bad for your health? And why is red meat technically now considered a class 2A carcinogen that when you take it, in theory, increases the chance that if you have a certain genetic background, that you will develop colorectal cancer by a certain age. And I want you to start thinking about why this is important in terms of red meat and processed meat. I also want you to be thinking about it this way. Just because the World Health Organization made this proclamation, they had 800 papers that they reviewed, and they came to this conclusion. Is this actually right? Just because it's in a journal, is that actually correct? And I want you to think a little bit about this. Now, why is this important for us? In the North American diet over the last 40 years, red meat consumption has actually skyrocketed. In parallel to that, and it's not strictly causational, but correlational perhaps, and observational for sure. that along with this increase that we have in red meat consumption, we've also seen the rise of colorectal cancer. And some of you might argue, well, we're simply better at detecting colorectal cancer than we were before. Not true. Not true. The way that we detect colorectal cancer has not really changed that much in the last 40 years. So why is this sort of happening? In many ways, parallel to what we're seeing with metabolic syndrome and food intake, how is this happening? Why is this happening? And is this in fact accurate? So there's been this rise in colorectal cancer, and we're going to go through the mechanisms of why colorectal cancer occurs, and we're going to see if meat indeed does play a role in colorectal cancer progression. But when we're asking this, are these really related? Is it a correlational study? And should we actually be paying attention to the World Health Organization? So I want to start off with a really basic question. What is red meat? The definition, by the way, isn't universally agreed upon. And I'm using the definition that was agreed upon by that World Health Organization paper back in October of 2015. And red meat is simply looking at the meat and determining its color. And the color of the meat, if it's red, is often the best way to describe this. White meat, for example, often refers to meats that are pale before they are cooked. So things like poultry, fish, for example. Also, some pork is also considered to be white meat. And red meat, things like beef, lamb, also pork in some instances. And some people consider duck, which is a form of poultry, to be red meat. So it's really difficult. I'm going to use a simple operational definition that if it looks red, it's red meat. Okay? So nothing fancy, nothing about its taste, nothing about how it gets prepared, nothing about where it comes from. Some people look at it as a category of mammalian meat products, etc., and game, sort of a third category. I'm just going to operationally define it as the color of the meat before it becomes cooked. So again, I won't go into a lot of details related to this and whether or not there's any difference in flavor associated with certain types of meat versus others. But we're simply going to be looking at red meat, and we're going to eventually look at processed meat as we go along. Now, I want you to keep in mind one thing, or a couple of things as we go along, and this again being the last lecture that you need to worry about for your midterm. Red meats generally, if you've taken a physiology course at some point in time, you'll know that different muscle fibers exist. And we have slow oxidative, fast glycolytic fibers, all of those different things. That we know that some muscle fibers are more enriched in one particular protein known as myoglobin. And myoglobin has associated with it the globin chain, but also associated with it, it has another factor which is known as heme. And I'll come back to heme in a moment. But for the time being, I'm going to ask you to just keep in the back of your mind that red meats tend to have more of the protein known as myoglobin, okay? And all of you have taken some physiology course at some point in time, right? Second year physiology course like Bio 271 maybe, or PSL 300 or 301. So when we're going and taking a look at some of the papers, including Lancet Oncology, I want you to keep in mind a couple of things. A lot of our studies come from a lot of different sources. So there isn't sort of this universal mechanism that everyone agrees on, just like no one agrees really on what red meat is and what white meat is. When we talk about the overall effects of meat, some of it is from different types of studies. Just out of curiosity, how many of you would actually eat meat after I've just went through all of this with you? Oh, this is a bad thing. Anyone want a hot dog? Okay, whoever wants a hot dog, thanks, Nick. Oh, I don't want one, thanks. Everyone, please say thank you to Nick, who took the time to come all the way out here. Thanks, Nick. Appreciate it. Thank you. It's up to you. I'm not going to take any responsibility for any long-term health consequences, by the way. Now that we're talking about meat, and in fact, this is actually not a true variant of processed meat, but a form of meat. It's beef, so it is considered like red meat. No one wants to try it? They're just going to get colder and colder. Come on down. Brave individuals. Okay. So when we're looking when we're looking at these different types of model organisms, some of these experiments come from Drosophila, which is an invertebrate system. Some of these come from C. elegans. Some of these come from zebrafish. You have to keep all of these different things in mind. Again, some of these are purely observational studies. The study that I'm going to show you, and you'll read a little bit about for your tutorial in the form of a news article, are observational. Those 800 articles that they say and mention in that Toronto Star article are all observational studies for the most part. And when we start looking at how they do those biological studies, the ones that involve looking at the mechanisms that might be involved in cancer within the gut, a lot of these are based on cell culture. So again, what I want you to take away from all of this is that one of the cofactors in meat that gives it its red color, the red meat that we're talking about, is heme. Heme is found in things like blood. Heme is also found as a component of things like myoglobin, and that red meat color is really because of this component known as heme. And these hemoproteins have a lot of different roles in biology. One thing is we don't absorb heme proteins well through the small intestine. So in other words, other molecules like glucose and other things get easily absorbed from the small intestine. When you're eating red meat, the heme proteins that you're eating along with the meat stay in your gut all the way down to the large intestine. And one of the things that is really bad about this is that this heme, in theory, can actually penetrate through that mucus layer lining that we talked about several times now, and it can actually damage the surface epithelial cell. Now think about this. Heme protein, and one of the reasons why it can damage is because of the iron associated with it, can puncture your cells within the intestine. Now, does that make sense in terms of colorectal cancer? Maybe that's the first sort of hit or the first sort of irritation that starts this whole process off. And in order to respond to this, the body responds by hyperproliferation. That area that's been damaged because the gut firewall has been breached actually starts to hyperproliferate those stem cells. And here's an example for that I often use on tests. I talked to you about stem cells in passing when we talked about the gut firewall. What region of the gut do we normally find these stem cells in? Do you remember? In the crypts, that's right. So we would expect to see more migration, more proliferation in the crypts of the intestine and them moving up. And that is something that you should make sure that you are well aware of for the upcoming midterm. So this hyperproliferation of these stem cells in response to this damage caused by this red meat containing heme protein now causes your cells to start acting abnormally. And it causes hyperplasia, an overgrowth of tissue, which is that precursor to a condition which we're going to look at in more detail, that colorectal cancer. Do you know what precedes colorectal cancer? So you get this massive tissue growing in your gut. Do you know what that's called before it becomes a full-blown cancer? Could be a tumor, but these small stalk-like cells, these outgrowths, yes. A polyp, and you'll hear a little bit about polyps in the readings that you have. So you start off with these polyps, which then become this full-blown cancer. So you'll be reading a little bit more about this. I'm hoping that you'll start thinking about, why does meat actually cause cancer? Why is it bad for our gut? I've been hearing about this all this time, but I really had no idea that this is the reasons why. And so you're going to hear a little bit about the fact that red meat is now considered a class 2 carcinogen. In other words, it's likely a carcinogen. They don't have definitive observational proof yet. They haven't been able to dump this onto a group of cells and cause this direct conversion. Processed meats in that Lancet article were identified as a class 1 carcinogen. It definitely causes cancer. Not likely, it causes cancer. Just like smoking causes cancer, processed meats cause cancer. Okay, and we'll talk a little bit more about this on Monday as well. So this is in the same category as things like asbestos, air pollution causing cancer, viral infections like HIV causing cancer, taking in tobacco causing cancer, being exposed to X-rays causing cancer, alcohol causing hepatic cancer, meat causes colorectal cancer. And again, that was sort of the bad news. So we're going to come back to this in some more detail. These hemoproteins, these heme groups, actually contain a chemical known as N-nitroso, and you'll hear about N-nitroso on Monday in your tutorials. And these red meats also contain other chemicals in some cases that will generate even more of these N-nitroso compounds within the gut. So some processed meats, in addition to containing heme, also have preservatives which make this worse. More N-nitroso compounds get generated from the preservatives that are found in some red meats. Cooking at high temperatures, so if you're grilling, barbecuing, etc., can also create other chemicals in addition to these N-nitroso compounds that will elevate the risk of cancer as well. And in generally speaking, what makes this worse is that we tend to see this in red meats. We see N-nitroso compounds from the heme and the preservatives in red meat. We usually don't try to preserve white meat. Think a little bit about that. You either have like dried chicken breasts, but we don't try to create like beef jerky, for example, from chicken quite as often. So we'll come back to this eventually down the road. So this is the last lecture. This is the last of the material that you're going to need for your midterm. Now I'm going to troll you really, really heavily. So I want you to start studying for this. I haven't made the midterm yet. I'm creating the midterm this weekend. And so you're going to help me a little bit in making the midterm over this coming weekend. So I need a couple of volunteers to help me out with this process. Does anyone volunteer to help me out with that? You may or may not want to do this. Yes? Good? Yes? Okay. All right. So since she's here closer to the front, if you want to work your way up to the front. All right. If you can help me out, I need your help just a little bit. You're going to help me make the midterm exam. Yes. All right. Sorry? I'm not going to ask you any questions. Well, actually, I'm going to ask you this one question. Can you pick one of these? Just one.
Okay. Oh my gosh. Okay, I'll just take the middle one.
The middle one. Okay, and I feel like we're at the Academy Awards with that low diversity again. 3. So you. Oh, sorry, we did the wrong one. Never mind. Okay, come back. Do the blue ones. So this is for multiple choice.
Okay, okay.
Okay, here we go back. 26 multiple choice. Okay. All right. 26. All right. So we'll have 26 multiple choice test questions on your midterm. Okay. So yeah. Sure. Oh. Yeah. Short answer questions. Anyone want to pick the short answer questions? Yeah. Let me just. You want to help me out here? And here we have 3. By the way, because I'm trolling you, so you have 3. You have 3. And actually, you did actually have a choice of 2. Oh, yes. Yeah, I'm kind of like that. Okay. The last one. Anyone want to do it before I put the slide up? Oh, everyone's scared now. No one? You want to try it? Okay. Tearful. Okay, before you start picking, Cheerful's in here somewhere. Oh.
This second one is no, no. Oh, this one.
Oh, okay. All right, great. All right, thank you. Um, so I will be posting this. Let me give you a little context to this. You will have 26 multiple choice test questions. Some of them will be a little longer, some will be a little shorter. I'm very conscious of the fact that you only have 50 minutes to write this. Of the three questions, one will simply be labeling a diagram. So it won't take you very much time to do that. And one of those short answer questions will also involve you providing some definitions. Again, really short, 1 or 2 sentences. I'm not trying to get you to write a whole page essay or anything else. Will it be fair? That's something that you'll have to tell me after the 26th, but I'm going to try my best to make it fair. And because that probably left a bitter taste in most of your mouths, I guess, that whole process, this is the last of our fiber for this week, and we're moving on to other things. But if you want to help yourselves with a little bit of good food, please help yourselves as well. I'll see you all on Monday. Please make sure you read the really short article. That's the only article that you have this week related to your tutorial. Thanks, and see you all Monday.